to minimize self-ligation of vector and reduce background colonies. The dephosphorylation reaction requires the hydrolysis of the ester bond between the terminal 5' nucleoside and the phosphate group. This is achieved using a family of enzymes known as phosphatases. Phosphorylation can be accomplished with any number of phosphatases, including recombinant shrimp alkaline phosphatase, or RSAP. Visit chromathenb.com for the full list of products available for this application.
DNA ligation is the formation of a covalent bond between adjacent DNA fragments, frequently a vector and a gene of interest. Ligation is usually the final step before transformation in the cloning workflow. DNA ligase may be used to join double-stranded DNA fragments with either blunt or cohesive ends to form recombinant DNA plasmids. DNA ligase catalyzes the formation of a phosphodiester bond between juxtaposed 5' phosphate and 3' hydroxyl termini in duplex DNA. Ligation proceeds in 3 steps. Initially, the ligase is self-adenylated by reaction with free ATP, or NAD. Next, the adenyl group is transferred to the 5' phosphorylated end of the donor strand. Lastly, the formation of the phosphodiester bond proceeds through the reaction of the adenylated donor end with the adjacent 3' hydroxyl acceptor, accompanied by the release of AMP. While T4-DNA ligase is the most commonly used ligase for cloning, there are several options from NAD to choose from, including convenient master mix formats. For help with selection of a DNA ligase optimized for a specific end type and application, view our selection chart at NADsticktogether.com. Visit clonewithnad.com for a full list of products available for cloning.
Golden gate assembly is a method for assembling multiple DNA fragments relying on the activity of type 2S endonucleases that cleave outside of non-palindromic recognition sites. In this example, the destination vector is designed to include BSA1 recognition sequences in opposing directions to allow for the assembly of the desired fragments. Using PCR, BSA1 recognition sites are incorporated into the fragments in the proper orientation through primer design. If the destination vector with BSA1 flanking sequences is not available, BSA1 sites can similarly be introduced into any plasmid sequence by PCR to make a linearized form of the destination vector. In either case, the PCR-amplified fragments are ready to be assembled into the destination vector in a single-tube reaction containing BSA1 and T4 DNA ligase. Cleavage with BSA1 exposes complementary sequences for fragment assembly. A final, short 55 degrees Celsius step at the end of the assembly reactions favors digestion, ensuring only linear DNA remains. BSA1 sites, and therefore only the plasmids with the correct insertion will remain intact and ready for the last step, which is transformation. Insert fragments can also be pre-cloned into plasmids flanked by BSA1 sites. During assembly, inserts in vector will ligate to yield the final assembled product, which lacks BSA1 sites. Again, only the plasmids with the correct insertion will remain intact and ready for transformation. For a list of other type 2S restriction enzymes that can be used for Golden Gate assembly or other applications, please visit www.ned.com/type2s table. One of the unique features of any builder hi-fi DNA assembly master mix is the ability to remove 3' and 5' end mismatch sequences upon fragment assembly. During fragment assembly, T5 exonuclease will remove the bases on the 5' end, generating a 3' overhang. Single stranded 3' ends can then anneal. The DNA polymerase fills the gap, and DNA ligase joins the adjacent fragments. But what happens if there are extra bases on the end of your 3' end that you do not want to incorporate into your assembly product? Any builder hi-fi is capable of efficiently removing 3' end mismatches, up to 10 base pairs from the 3' ends, so that they can be efficiently joined together. In this example, we want to join a linearized vector in an insert at the 5' position of this restriction site. The insert is generated by PCR, and it has overlaps corresponding to the vector. When the overlaps anneal during the assembly process, there is a mismatch at the 3' end of the vector. By proofreading, DNA polymerase removes the extra bases at the 3' end. The polymerase fills the gap, and DNA ligase seals the remaining nick. Unlike other cloning methods, any builder hi-fi allows you to digest the restriction enzymes and conveniently assemble your DNA fragments at any position.
Restriction enzymes identify a specific recognition sequence in the DNA and generate double-stranded breaks in the DNA duplex. Cleavage results in fragments with either a 5' or 3' overhang, commonly referred to as sticky ends, or no overhang, referred to as blunt ends. The 5' phosphates and 3' hydroxyls are maintained after cleavage, leaving the DNA ends ready to be joined via ligation. The ability of some restriction enzymes to predictably cleave DNA and generate distinct DNA bands with ligatable ends has made them an invaluable tool for recombinant DNA technologies. When selecting restriction enzymes for use in a cloning experiment, it is important to determine which enzymes will produce ends compatible with the selected vector, confirm that recognition sites do not occur within the DNA fragments to be cloned, examine the methylation sensitivity of your selected enzyme to confirm that host methylation by DAM and/or BCM methylases will not block cleavage. NAD offers a variety of online interactive tools to aid in selecting which enzyme to use, as well as parameters for setting up reactions. Visit clonewithnad.com for a full list of products available for this application. Visceral cloning evolved from the discoveries of site-specific nucleases and DNA ligases in the 1970s to enable the era of molecular cloning. At the beginning, cloning a gene into a plasmid allowed the host organism to replicate the cloned sequence, and this procedure fueled the early gene sequencing, protein expression, and gene function studies. The first step is to identify unique restriction sites in your source DNA that can be used to isolate the fragment of interest. We recommend using NAD Cutter, an online tool available at nadcutter.nad.com. In many cases, PCR can be used to add the necessary restriction sites to the gene of interest to facilitate directional cloning. Mr. Vector and DNA fragment of interest with a single restriction enzyme for non-directional cloning, or a pair of restriction enzymes possessing different cleavage sites for directional cloning. The use of two different enzymes that produce blunt ends creates a non-directional cloning strategy. If you are using a single restriction enzyme or two enzymes that produce blunt ends, additional screening may be required. To avoid the possibility of vector subligation in the ligation step, it is common practice to dephosphorylate the vector to remove the 5' phosphate group that the DNA ligase means for phosphodiester bond formation. Depending on how you generate your vector and insert, other end treatments may be required. These include blunting, A-tailing, and phosphorylation. Select a ligase to covalently join the vector and insert. Transform the recombinant plasmid into competent E. coli. Spread onto agar plates that contain the appropriate antibiotic for selection. Screen the resulting colonies for the gene or fragment of interest. Although the basic workflow in traditional cloning had not changed much since the 1970s, the introduction of faster and much more robust enzyme and vector combinations, as well as wider specificities in the last decade, has made this approach easier and faster than ever before. Visit clonewithnad.com for a full list of products available for this application. PCR cloning is a common approach for the cloning of any DNA fragment of interest. This approach does not require a lot of input DNA and allows for the cloning of fragments for which the whole sequence may not be known. By amplifying the DNA fragment of interest with a polymerase using primers that are complementary to the DNA sequence of interest. With each cycle, the resulting DNA is amplified exponentially. The resulting amplified DNA will have either a single A overhang if Taq DNA polymerase was used, or a blunt end if a high-fidelity polymerase, such as Q5 high-fidelity DNA polymerase, was used. Next, join the amplified DNA fragments to the appropriate vector. This can be done by the use of the DNA ligase, or by use of activated vectors that are covalently bound to an effector enzyme, which facilitates vector-to-insert joining. Depending on the vector chosen, the fragment of interest may be inserted non-directionally at the ligation junction, such as in PA cloning. Some supplied vectors can contain a toxic gene. The use of toxic gene fusions ensures that only competent cells that have taken up the vectors with successful insertion of the target gene sequence survive, as the insertion disrupts the expression of the toxic gene. These vectors are known as suicide vectors. Form the recombinant plasmid into competent E. coli. Spread onto agar plates that contain the appropriate antibiotic for selection. Screen the resulting colonies for the gene or fragment of interest. In recent years, the introduction of fast and robust high-fidelity polymerases has made this approach easy and reliable. Visit clonewithnad.com for a full list of products available for this application. Building upon our introduction to Gibson assembly, which detailed the versatility and power of the Gibson assembly master mix, we will now walk through the protocol for preparing fragments for assembly using either Gibson assembly master mix or the Gibson assembly cloning kit. It is advisable to begin by assembling the final sequence in silico. This enables visualization of fragment junctions and facilitates the choice and design of the overlapping primers. Design the nucleotide overhangs, which will enable correct fragment annealing. For optimal assembly, we recommend using 15 to 30 base pair overlaps that exhibit a melting temperature greater than 48 degrees Celsius. The basic steps of fragment assembly are as follows. First, design primers for your fragments. The primers will be designed to include a 15 to 30 base pair overlap with the primer sequence on the complementary strands. Additional primer design approaches include adding the overlap region to the forward primer of fragment B, or splitting the overlap region between the reverse primer of fragment A and the forward primer of fragment B. After PCR, the resultant fragment includes the overlap region and is now ready to be joined during Gibson assembly. These same basic steps can be used to clone an insert, such as a LAC V, into an expression vector like PET21A using PCR. You will generate 4 primers, 2 forward primers, and 2 reverse primers. To design and overlap the sequence, chain is formed in the penile C5X vector after digestion with NCR1 and SBF1. The overlap region of the forward primer for the gene of interest should line upwards with 3' end of the overhang and extend back until the melting temperature of the overlap is greater than 48 degrees Celsius. To ensure preservation of the reading frame, nucleotides may be added between the overlap region and the gene-specific sequence. A similar approach is then taken with the reverse primers. After viewing these examples, you should now have an understanding of how to design primers to enable fragment assembly with Gibson Assembly Master Mix or the Gibson Assembly Cleaning Kit. The Gibson Assembly Cleaning Kit, which includes both Gibson Assembly Master Mix and any of these 5 alpha confidence cells, enables the assembly and cleaning of multiple fragments into any vector in just under 2 hours. Gibson Assembly, developed by Dr. Daniel Gibson and his colleagues at the J. Craig Venter Institute, is an effective method for the assembly of multiple DNA fragments. This is accomplished in a single-tube isothermal reaction with Gibson Assembly Master Mix. The method utilizes adjacent DNA fragments with complementary ends, which can be added, for example, by PCR. Then, the overlapping fragments are added to the Gibson Assembly Master Mix and incubated for 1 hour at 50 degrees Celsius. During the 1-hour incubation, the Master Mix's 3 enzyme activities set to work on the fragments. First, a 5' to 3' exonuclease activity creates single-stranded 3' overhangs. These complementary sequences then anneal, creating the double-stranded DNA of interest. DNA polymerase then extends the 3' end, filling the gap. Since DNA ligase seals the remaining mix. The end result is a fully sealed double-stranded DNA molecule that can serve as template for PCR, RCA, or a variety of other molecular biology applications, including direct transformation. Gibson assembly has been successfully used to reliably join up to 6 DNA fragments into a single